start our discussion with the bacterial cell division. So bacteria divide. The division normally occurs by binary fission. So sometimes cell division takes place by the process called as budding, but budding is mostly common in the yeast cells or in the fungal cells. So otherwise, bacteria most commonly divide by the process named as binary fission. So what happens in binary fission? Basically, the bacterial cell duplicates everything. So it starts duplicating the cellular constituents, the cellular organelles, as well as the genetic material. So the chromosome starts dividing as well. Once the chromosome has been completely divided, there is a stage or step known as a cell elongation. So bacterial cell lengthens or it increases in size, and then after that a process called septum formation starts. So a septum is just the generation of a new wall between two divided chromosomes. Once the septum formation is completed, that means all the organelles which were duplicated are now divided between the two cells, and then last stage will be cell separation. So once septum formation completes, two cells get separated. So two cells are generated from one mother cell. Okay, so that is why this process is known as binary fission. So this is the common way or common mode of reproduction for bacteria. So for the matter of fact, each square centimeter of skin hosts an average of 1 lakh organism, each square centimeter. And bacteria reproduce so quickly that their population is restored within hours after washing. Restored to 1 lakh, around 1 lakh after washing. And that is why, as we say, it is very, very important to wash our hands repeatedly. Now, phases of bacterial growth. Now, you have learned about the preparation of media and inoculation of that media during your practicals. Now, consider that you have a test tube of nutrient broth, and in that nutrient broth, in that fresh nutrient media, you have inoculated or you have added few organisms, one loop full of organisms, microorganisms, suppose E. coli. Now, what happens to that E. coli when you freshly add it into a nutrient material or nutrient media? So that organism, that population of organism will undergo 4 distinct phases of growth. So those phases are named as: first phase is the lag phase, second phase is log phase or exponential phase, third phase is stationary phase, and the last one is decline phase or death phase. Now, all these phases together, if put one by one, one after the other, they form a curve, and that curve is known as standard bacterial growth curve, which can be seen in the next slide. So as you can see, the growth pattern starts at the lag phase, it continues in the log phase, then it goes on into the stationary phase, and then last phase is decline phase or death phase. Just observe one thing in this graph. On y-axis is labeled as logarithm of number of cells. Just keep that in mind for a while. And x-axis is, of course, time. Okay. So now let's understand each phase one by one. So lag phase. What happens in lag phase? So when you freshly introduce the organism in a fresh nutrient-rich media, what happens? That organism will start growing. But growing in what terms? That organism will grow in size. So the size of the bacterial cell will become larger, and in the lag phase, bacterial cells are involved in synthesizing the cellular components or building blocks of the cell, which are required for further growth and cell division. So in lag phase, bacteria particularly does not increase in number, it increases in size. So they are metabolically active, they grow in size, they keep on synthesizing enzymes and other material, other components, and they generate they produce large quantities of energy in the form of ATP, which is stored inside the cell, and it is required further for the next phases of the growth. So after lag phase, bacterial population, cell population goes into the log phase or exponential phase. As you can see, there is a sharp increase in the number of cells. So what is happening here? Bacteria is now increasing, but not just in size, also in the number. So basically, cells are rapidly dividing and more number of cells are getting generated. That is how number of cells will increase rapidly in log phase, or it is also known as exponential phase. So how does this increase is explained? That increase is happening at a regular genetically determined time interval, and the time interval is called as generation time. Okay, so the population of organisms doubles in each generation time. For example, suppose, just for an example, E. coli, if for E. coli, the generation time say is 2 minutes. Okay, and to start with, the population of E. coli was 2 cells. So what will happen after 2 minutes? The E. coli will divide and the population will double. So after 2 minutes, the population will become 4. After 4 minutes, population will become 8. After 6 minutes, population will become 16. After 8 minutes, population will become 32, and so on. So generation time is the time interval, which is, again, I am repeating this, genetically determined for each species, and in that time interval, the population of organisms doubles in number. Okay, so it is specific to individual species. So it differs from species to species. So that was about log phase. In log phase, bacterial cells rapidly increase in number. So log phase or exponential phase is followed by stationary phase. So you can imagine what is happening here. The nutrient broth test tube is same as we started. So what is happening now? Bacterial cells are multiplying very rapidly. The cell number is increasing very rapidly, but the amount of nutrients present in that nutrient broth test tube is limited. So what will happen as a number of organisms will start growing, there will be lack of nutrient supply. So simultaneously, cells will start aging and dying from lack of nutrients. So what is happening in stationary phase? The total number of organisms is kind of remaining fixed or constant, because cells are getting generated, cells are multiplying, but at the same time, approximately equal amount of cells are dying as well. So that is how the bacterial population enters in the stationary phase. What happens after this? After this, the population starts declining. Okay, it is known as a decline or death phase. So basically, cells lose their ability to divide, and there is nutrients are now finished. There is nothing to eat for the cells. So cells will gradually start dying. Okay, so both log phase and death phase or decline phase are logarithmic. In a log phase, cells increase in exponential or logarithmic manner, and in death phase or decline phase, cells decrease in the exponential or logarithmic manner. So this whole, these 4 phases form this whole curve, which is known as bacterial growth curve. Now next, after this, you have to solve this problem. So there is one question for you. Just note this down in your notebooks and try to solve this. You have to plot a graph for given data. On x-axis, you have to put time in minutes. On y-axis, you have to plot number of cells. The generation time for the given population is given as 1 minute, and the number of cells or initial population is given as 10 cells. So use this data and try to plot a graph for this data. After this, let's continue with the nutritional requirements or nutrients required by the bacteria for growth. So there are two types of nutrients which are required by bacteria. These are categorized as macronutrients and micronutrients. So macronutrients are basically required in large amounts, in large quantity, and micronutrients are required in trace amounts or very small quantities. So the macronutrients, there are four macronutrients: carbon, nitrogen, sulfur, and phosphorus. And there are number of other elements which are required in very small quantities. They are known as micronutrients. So let's start with carbon first. So carbon basically acts as a building block for many components of the cell. Bacteria can obtain this carbon from many organic sources as well as from glucose present in the nutrient media, by fermentation, or by the process known as Krebs cycle. So bacteria can also synthesize many cell components from the intermediates of the pathways of cell Krebs cycle. So carbon is one of the major source of nutrient which is required as a building block for the growth and division or multiplication of bacterial cell. Now, next macronutrient is nitrogen source. So all organisms, including microorganisms, require nitrogen. Nitrogen is basically required for synthesis of various enzymes, amino acids, proteins, and nucleic acid. So microorganisms can obtain this nitrogen from inorganic sources, or few organisms also obtain energy by metabolizing inorganic nitrogen containing substances. So once amino acids are synthesized or obtained from medium, they can be used in the protein synthesis. So nitrogen acts as a vital component for the final process of protein synthesis. Now, next components are sulfur and phosphorus. These are two other macronutrients which are required in larger amount. So these are very, very important cell components, and bacteria can obtain sulfur from inorganic sulfate salts and from sulfur containing amino acids. And sulfur is basically used to make proteins, coenzymes, and other cellular components. As far as phosphorus is concerned, phosphorus is obtained mainly from inorganic phosphate ions, and phosphorus is involved in synthesis of ATP, phospholipids, and nucleic acid. So those were the four macronutrients: carbon, nitrogen, sulfur, and phosphorus. Now, other minerals are required in very, very small quantities, very, very small amounts. Those are known as trace elements. So examples of such minerals are copper, iron, zinc, cobalt, etc. So these are involved, they serve basically as cofactors in various enzymatic reactions, are involved and are also involved in the synthesis of vitamins for the cell growth. Another important constituent is calcium. Calcium is required by gram-positive bacteria for synthesis of cell wall, as well as by spore-forming organisms for synthesis of spores. So that was about the nutritional requirement by bacteria, by organism to grow and multiply. So those were the macronutrients and micronutrients. So while preparing the media, nutrient media for bacteria, you have to make sure that you are adding all these ingredients, macro as well as micronutrients, as a component of the media. Then only it will serve as a nutritional supplement for the growth of organism. So we have discussed about the growth phases of the organism as well as the nutritional requirements. Now, next part, we are going to discuss about the physical parameters which affect the growth of bacteria. For example, the temperature requirement or the moisture requirement or the oxygen requirement, etc. And after that, we will learn about different types of media and how to prepare those media. So that's it for this lecture. Thank you.